Hi and Assalamualaikum. My name is Nabila Balkis binti Limon, SB 22001. Now I will present about our product related to biotechnology, which is a multivitamin. First, I will present about the overview of the project. The Envision project revolves around the establishment of a cutting-edge certified laboratory specializing in the quality control testing of biotechnology-related products with a focused emphasis on the meticulous analysis of the multivitamins. This comprehensive initiative is propelled by burgeoning demand for reliable and high-quality biotechnological products, aligning with the evolving landscape of consumer expectations, regulatory standards, and scientific advancements. Now, I will talk about our objectives. The goal of this biotechnology initiative is to establish a certified laboratory dedicated to quality control testing for multivitamin biotechnology products. The aim is to implement precise and reliable testing methods that align with industry standards. So, why is validation important to our product? Validation is crucial to confirm the accuracy, precision, and reliability of the testing process. It instills confidence in stakeholders, including manufacturers, regulatory bodies, and consumers, ensuring that the laboratory consistently produces dependable results for multivitamin products.
Hello, my name is Sofena Binti Arifin, and today I will discuss the assignment's introduction. Briefly put, it's about in the world of biotechnology, people want safe and high quality products. This is super important for things like vitamins in commercial product because they show how good and effective the biotech stuff is. To make sure everything is safe and top notch, we have to be really careful and check things a lot.
Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Afina Nabiha binti Asani and I'm going to present about the defined method scope. The first is the sample preparations. Matrix type is usually tablets, powder, liquid and injections. For homogenizing is by mixing and blending each of the vitamins and minerals. The dilution factor for a multivitamin solution is a measure of how much the original concentrations of the solution is reduced when it is mixed or diluted with another substance, typically a liquid like a water or juice. The dilution factor is expressed as a ratio is calculated by dividing the final volume of the diluted solutions by the initial volume of the concentrated solutions. For sample extractions, extraction solvents for multivitamins can include organic solvents such as hexane, ether, dichloromethane, and chloroform, which are widely used for the extraction of fat-soluble vitamins. Extraction times. Study mentioned that the extraction time for fat-soluble vitamin is 5 minutes. For temperature control, the content of vitamins will gradually increase when the temperature increases. The next part is standard preparations. Certified reference material available in the form of standard reference material in 3,289 multivitamin tablets, which was developed by the National Institute for Standards and Technology in collaborations with the other organizations. And the last step is separations. Usually used is the reverse phase high performance liquid chromatography and also the ion pair reverse phase high performance liquid chromatography.
Greetings, Doctor. My name is Nimalan, SB22049. So now that we have taken a look at the method scope definition, let's take a look at the validation criteria definition. So there are 6 of them, which are specificity, linearity, accuracy, precision, and also finally limit of detection and limit of quantification. So let's take a look at them one by one. First criterion is specificity. So specificity is used to establish quantitative measures and performance standards and also used to ensure that the analytical method can accurately and selectively determine the concentration of individual multivitamins. Next is linearity. Linearity ensures that the response of the method is directly proportional to the concentration of analytes over a specified range. This refers to the accuracy and reliability of the analytical methods used for determining the presence and quantities of various vitamins in the multivitamin formulation. One is accuracy. The validation criteria of multivitamins in terms of accuracy refer to the ability of analytical methods used for determining the presence and quantities of various vitamins in the multivitamin formulation to provide precise and correct measurements. One key aspect of accuracy is the biasness. The method should have minimal or no systemic errors, ensuring that the measured values are accurate representations of the actual vitamin content in the sample. Fourth criterion is precision. Precision refers to the ability of the analytical methods used for determining the presence and quantities of various vitamins to provide consistent and reproducible measurements. The key aspects for precision are repeatability, reproducibility, and also intermediate precision. Final criterion is limit of detection and limit of quantitation. Limit of detection, in this case, refers to the lowest concentration of a vitamin that can be reliably detected but not necessarily quantified. Meanwhile, limit of quantitation refers to the lowest concentration of vitamin that can be accurately measured and quantified with acceptable precision and also accuracy.
Now, I will talk about method validation plan. Firstly is specificity. Specificity is ensured by analyzing blind samples, spike samples, and actual product samples to confirm the method's accurate detection and quantification of targeted vitamins. Secondly is linearity. Prepare a series of standard solutions covering the expected concentration range. This involves creating a set of solutions with known concentrations that span the range of concentrations expected in the samples. Analyze each standard in three decades. Each standard solution is analyzed three times independently to ensure consistency and reduce the impact of any experimental variability. Construct calibration curve. Utilize data from standard analyzers to create a calibration curve, mapping measured concentrations against known concentrations. This curve acts as a reference for determining unknown concentrations in subsequent samples. Assess linearity with statistical methods. Apply statistical techniques to evaluate the linearity of the calibration curve, ensuring the method delivers accurate and reliable results across the entire concentration range. Next is precision. Precision is evaluated through the analysis of replicates for repeatability and intermediate precision, ensuring consistent and reliable results within a single analysis and across different conditions. Last but not least is accuracy. Accuracy is verified by spiking product samples with known vitamin amounts and comparing measured concentrations with expected values, validating the correctness of the method in practical applications. Next, I will pass this to the next speaker.
My name is Lim Meng Chong, SB22061. Next, my group are going to present about performance test. For our group performance test, there's going to be three types, which is randomized control trials, biochemical measures, and cognitive performance tests. Firstly, randomized control trials, which are called RCTS. These are considered the gold standards for testing the efficiency of interventions like multivitamins. RCTS can measure various health parameters, including cognitive functions, physical performance, and disease risk markers. These studies follow groups of people over time to observe their health outcomes in relation to their multivitamin use. While observational study cannot establish cause and effects relationships like RCTS, they can provide valuable insights into the real-world use of multivitamins and their potential associations with health. Second one is biochemical measures. Researchers can measure levels of certain biomarkers in the blood or tissue that may be affected by multivitamin use. For example, C-reactive protein is a marker of inflammation, and studies have shown that multivitamin may reduce its levels. Measuring blood levels of vitamins and minerals before and after multivitamin supplementation can assess whether the supplement is effectively raising nutrient concentrations. For the third one is cognitive performance test. This test assess various cognitive functions such as memory, attention, and executive function. They can be used to measure whether multivitamins improve cognitive performance in individuals with or without cognitive declines. Techniques like functional MRI can be used to measure brain activities before and after multivitamins supplementation. This can provide insight into how multivitamin may affect brain functions. So I would like to pass to the next speaker.
Among the questions that can be asked during ongoing routine tests is what is the purpose of the system sustainability test? So the answer is to assess and guarantee the multivitamin product's sustainability while taking into account elements such as long-term viability, ethical sourcing, and environmental effect. So the second one, why is the system sustainability test is necessary? So, to encourage multivitamin production and consumption that adheres to ethical and environmental standards and is responsible and sustainable. Then, who are the key participants in the system's sustainability test? There are manufacturers, government agencies, environmental groups, customer, and independent certification bodies. Furthermore, where should the system sustainability test be conducted? To be carried out at several phases, including procurement of raw materials, production, delivery, and post-consumer usage. It is important to conduct evaluation on a regular basis to guarantee sustainability. Where does the system sustainability test take place? The complete supply chain, including the procurement of raw materials such as vitamins and minerals, production plants, distribution system, and possible disposal location. And the last one is, how is the system sustainability test conducted? A number of variables are evaluated in the test, including the effect production has on environment, the ethical sourcing of ingredients, waste management, energy use, and compliance with sustainable certification and standards.
Hi everyone, my name is Isham SB206 that I present the system sustainability test and quality control and measurement. One crucial aspect of sustainable vitamin production is the ingredient source. Responsible companies prioritize obtaining raw materials from eco-friendly and ethical sources. This not only ensures the quality of the supplement but also reflects a commitment to environmental and social responsibility. Manufacturing processes play a significant role in the sustainability of the vitamin production. Companies adhering to sustainable practices focus on minimizing waste, reducing energy consumption, and adopting environmentally friendly manufacturing methods. Certifications such as ISO 14001, which addresses environmental management, are indicative of a commitment to sustainable manufacturing. Packaging is another critical consideration. Sustainable packaging practices involve using recyclable material, minimizing packaging waste, and adopting eco-friendly packaging solutions. The emphasis is on reducing the environmental impact associated with the entire product life cycle. Third-party certification provides a consumer with a reliable way to identify sustainable products. Certifications such as Rainforest Alliance, Fair Direct, and USDA Organic are indicators of adherence to strict sustainability standards. This certification assures consumers that the products meet specific criteria related to environmental and ethical considerations. Transparency is key in the sustainable supplement industry. Brands committed to sustainability are transparent about their supply chain, manufacturing practices, and overall environmental impact. This transparency builds trust with consumers and allows them to make informed choices aligned with their values. For the quality control in measurement, first, ethical quality control programs. This program chooses accredited labs and carry out accepting testing to assess the true nutritional content of goods. Besides, National Institute of Standard and Technology standard reference material. To verify the accuracy of measurements, it offers verified values of vitamins and other elements included in the dietary supplements. Third, dietary supplement laboratory quality assurance programs. With the help of this program, testing facility of dietary supplements may increase the consistency of their measurements, and National Institute of Standard and Technology can evaluate their effectiveness. Furthermore, micellar electrocardiogram chromatography. This method ensures the precision and dependability of the measurements by analyzing vitamin D2 multivitamin pills. Last, collaborative study. In the dietary supplement sector, multi-laboratory studies are carried out to assess the precision, concordance, and accuracy of measurements.
In conclusion, setting up a certified lab to test multivitamin product is super important for making sure they are safe and effective. It shows a commitment to quality, meeting rules and aims to build trust with customers, healthcare pros and regulators by using top-notch equipment, skilled staff and strict testing rules. Their certification is like a mark of quality, making people trust multivitamins more. That's all for our group. Thank you.